Explore CRISPR gene editing, how a bacterial immune system became a programmable tool, how Cas nucleases and guide RNA precisely edit genomes, and what applications, technical limits, and ethical challenges shape medicine, agriculture, and diagnostics today. Foundations. From bacterial repeats to a genome editing tool, CRISPR began as a curious pattern of repeated DNA discovered in 1987 and later recognized in bacteria as an adaptive immune memory. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. In 2012, Jennifer Doudna and Emmanuel Charpentier adapted the bacterial system into a programmable genome editing tool, earning the 2020 Nobel Prize. The system uses short RNA sequences derived from previous invaders to recognize matching DNA. In bacteria, Cas proteins, endonucleases cut viral DNA. Key components are the CRISPR array, guide RNA, and Cas nuclease. Typical guide RNAs are approximately 20 nucleotides long. This bacterial origin explains its precision, modularity, and wide uptake in labs globally since 2013. Mechanism: how Cas nucleases and repair pathways produce edits. At its core, CRISPR-Cas9 is programmable DNA scissors. A single guide RNA, sgRNA of about 20 bases, directs Cas9 to complementary genomic DNA adjacent to a short PAM motif. For SP-Cas9, NGG, Cas9 induces a double-strand break. The cell repairs this break via non-homologous end joining, NHEJ, or homology-directed repair, HDR. NHEJ is fast and error-prone, typically introducing insertions/deletions, indels that disrupt genes. Reported indel rates commonly exceed 50 to 90% in cell culture. HDR can introduce precise changes but is much less efficient, often under 1 to 20% depending on cell type and timing. Modern improvements include high-fidelity Cas9 variants and base editors that avoid double-strand breaks. Applications: clinical, agricultural, and diagnostic examples. CRISPR's versatility has driven diverse real-world efforts. In medicine, companies such as Vertex/CRISPR Therapeutics and Intellia are running clinical programs. Vertex CRISPR's Exacel targets sickle cell disease and beta thalassemia, while Intellia's in vivo NTLA-2001 targets transthyretin amyloidosis. Dozens of trials began after 2018 exploring cancer, blood disorders, and eye diseases. In agriculture, gene-edited crops and livestock, such as disease-resistant plants and hornless cattle, have been developed to improve yield and welfare without foreign DNA in some cases. In diagnostics, CRISPR-based tests, Sherlock detector, enable rapid nucleic acid detection and were adapted during the COVID-19 pandemic for point-of-care assays. Regulatory pathways vary by country, shaping adoption timelines. Advanced topics and implications: delivery, new editors, and ethics. Advanced challenges and innovations determine CRISPR's next decade. Delivery into human tissues is a primary bottleneck. AAV vectors have 4.7 kilobase payload limits, complicating delivery of large nucleases like SP-Cas9, 4.2 kilobytes coding sequence, whereas lipid nanoparticles, LNPs, offer transient delivery and enabled Intellia's in vivo trial. Off-target edits remain a safety concern. Engineered high-fidelity nucleases and shorter guides can reduce unwanted cuts. New modalities, base editing 2016 and prime editing 2019, permit single base changes or small insertions without double-strand breaks, lowering indels. Ethical implications escalated after the 2018 germline editing incident, prompting international calls for rigorous oversight, transparent trials, and global governance frameworks. CRISPR transformed biological research by converting a bacterial defense into a modular, programmable editing platform. Understanding Cas mechanisms, repair pathways, delivery hurdles, and newer editors clarifies strengths and limits. Responsible deployment, rigorous trials, and international governance will determine whether CRISPR fulfills its promise safely and equitably.